When spending time with your own family, friends, and neighbors, you may have noticed that many traits run in families. For instance, members of a family may share similar facial features, hair color, or a predisposition to health problems such as diabetes. Characteristics that run in families often have a genetic basis, meaning that they depend on genetic information a person inherits from his or her parent. What if you wanted to figure out how genetic information is transmitted between generations? For instance, you might be curious how traits can skip a generation, or why one child in a family may suffer from a genetic disease while another does not. How could you go about asking these kinds of questions scientifically? An obvious first idea would be to study human inheritance patterns directly, but that turns out to be a tricky proposition. In this video, we'll see how a 19th century monk named Gregor Mendel instead uncovered the key principles of inheritance using a simple, familiar system, the P plan.
Johann Gregor Mendel, 1822 to 1884, often called the father of genetics, was a teacher, lifelong learner, scientist, and man of faith. It would be fair to say that Mendel had a lot of grit. He persevered through difficult circumstances to make some of the most important discoveries in biology. As a young man, Mendel had a difficulty paying for his education due to his family's limited means, and he also suffered bouts of physical illness and depression. Still, he persevered to graduate from high school and later university. After finishing university, he joined the Augustinian Abbey of St. Thomas in Brno, in what is now the Czech Republic. At the time, the monastery was a cultural and intellectual hub of the region, and Mendel was immediately exposed to new teachings and ideas. His decision to join the order, against the wishes of his father, who expected him to carry on the family farm, appears to have been motivated in part by a desire to continue his education and pursue his scientific interests. Supported by the monastery, he taught physics, botany, and natural science courses at the secondary and university levels.
In 1856, Mendel began a decade-long research project to investigate patterns of inheritance. Although he began his research using mice, he later switched to honeybees and plants, ultimately settling on garden bees as his primary model system. A model system is an organism that makes it easy for a researcher to investigate a particular scientific question, such as how traits are inherited. By studying a model system, researchers can learn general principles that apply to other hard-to-study organisms or biological systems, such as humans. Mendel carried out his key experiments using the garden pea, Pisum sativum, as a model system. Pea plants make a convenient system for studies of inheritance, as they are still studied by some geneticists today. Useful features of peas include the rapid life cycle and production of lots and lots of seed. Pea plants also typically self-fertilize, meaning that the same plant make both the sperm and egg that come together in fertilization. Mendel took advantage of this property to produce true breeding pea lines. He self-fertilized and selected peas for many generations until he got lines that consistently made offspring identical to the parent. Pea plants are also easy to cross or mate in a controlled way. This is done by transferring pollen from the anthers, male parts of a pea plant of one variety to the carpel, the female part of a mature pea plant of a different variety. To prevent the receiving plant from self-fertilizing, Mendel painstakingly removed all of the immature anthers from the plant's flowers before the cross. Because peas were easy to work with and prolific in seed production, Mendel could perform as many crosses and examine many individual plants, making sure that his result was consistent, not just a fluke, and accurate, based on many data points.
Mendel experimented on pea plants and considered 7 main contrasting traits in the plants. Seed shape, dominant trait, round, recessive trait, wrinkled. Seed color, dominant trait, yellow, recessive trait, green. Flower color, dominant trait, purple, recessive trait, white. Flower position, dominant trait, axial, recessive trait, terminal. Plant height, dominant trait, tall, recessive trait, short. Pod shape, dominant trait, inflated, recessive trait, constricted. Pod color, dominant trait, green, recessive trait, yellow.
Mendel's experimental setup. Once Mendel had established true breeding pea lines with different traits for one or more features of interest, such as tall versus short height, he began to investigate how the traits were inherited by carrying out a series of crosses. First, he crossed one true breeding parent to another. The plants used in this initial cross are called the pea generation or parental generation. Mendel collected the seeds from pea generation cross and grew them up. These offspring were called the F1 generation, short for first filial generation. Filius means son in Latin, so this name is slightly less weird than it seems. Once Mendel examined the F1 plants and recorded their traits, he let them self-fertilize naturally, producing lots of seed. He then collected and grew the seeds from the F1 plants to produce an F2 generation, or second filial generation. Again, he carefully examined the plants and recorded their traits. Mendel's first experiments. Mendel crossed purple flowers with white flowers. The offspring from this cross is called first-generation plants, or F1. All were purple. Where did the white flower trait go? Mendel's second experiments. Mendel allowed first-generation plants to self-pollinate. The recessive trait for white flowers reappeared in the second generation.
In 1865, Mendel presented the results of his experiments with nearly 30,000 pea plants to the local Natural History Society. Based on the patterns he observed, the counting data he collected, and a mathematical analysis of his results, Mendel proposed a model of inheritance in which characteristics such as flower color, plant height, and seed shape were controlled by pairs of heritable factors that came in different versions. One version of a factor, the dominant form, could mask the presence of another version, the recessive form. The two paired factors separated during gamete production such that each gamete, sperm, or egg randomly received just one factor. The factors controlling different characteristics were inherited independently of one another. Gregor Mendel, through his work on pea plants, discovered the fundamental laws of inheritance. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. Mendel conducted two main experiments to determine the laws of inheritance. These experiments were 1. Monohybrid cross experiment, and 2. Dihybrid cross experiment.
Monohybrid cross experiment. In this experiment, Mendel took two pea plants of opposite traits, yellow seed and green seed, and crossed them. He found the first generation offsprings were 100% yellow seed and called it F1 generation. Then, he self-pollinated F1 generation and obtained both yellow and green seeds in the ratio 3:1. Mendel even conducted this experiment with other contrasting traits like tall versus short, round versus wrinkled, etc. In all the cases, he found that the results were similar. From these, he formulated the laws of segregation and dominance.
Law of dominance. This is also called Mendel's first law of inheritance. According to the law of dominance, hybrid offspring will only inherit the dominant trait in the phenotype. The alleles that are suppressed are called as the recessive traits, while the alleles that determine the traits are known as the dormant traits. Law of segregation. The law of segregation states that during the production of gametes, two of each copies of each hereditary factor segregate so that the offspring acquire one factor from each parent. In other words, allele alternative from the gene pairs segregate during the formation of the gamete and reunite randomly during fertilization. This is also known as Mendel's third law of inheritance.
Dihybrid cross experiment. In a dihybrid cross experiment, Mendel considered two traits, each having two alleles. He crossed wrinkled green seed and round yellow seeds and observed that all the first generation, F1 generation, were round yellow. This meant that dominant traits were the round shape and yellow color. He then self-pollinated the F1 generation and obtained four different traits: wrinkled yellow, round yellow, wrinkled green, and round green seeds in the ratio 9:3:3:1. After conducting four other traits, the results were found to be similar. From this experiment, Mendel formulated the second law of inheritance, which is the law of independent assortment.
The law of independent assortment, also known as Mendel's second law of inheritance. The law of independent assortment states that a pair of traits segregates independently during gamete formation. As the individual hereditary factors assort independently, different traits get equal opportunity to occur together.
Although Mendel's work was largely accurate, his ideas were never recognized by his peers during his lifetime, 1822 to 1884. His findings were originally published in an obscure Austrian journal in 1866. He died in obscurity and his work went unnoticed until being rediscovered around 1900s by four scientists: the Dutch botanist Hugo de Vries, the German botanist and geneticist Karl Correns, the Austrian agronomist Erich von Tischer-Max-Seysenig as a graduate student, and the American wheat breeder and agricultural economist William Jasper Spillman. Farmers had known for centuries that crossbreeding animals or plants could favor certain desirable traits. As the 20th century dawned, these empirical observations could now be explained scientifically on the basis of the newly discovered ideas of Mendelian genetics. Mendel's pea plant experiments established by many of the rules of heredity, now referred to as the laws of Mendelian inheritance, which helped stimulate the rapid advances in genetics and plant breeding of the last century.